Welcome, everybody, to the February 2025 meeting of the Parrot Club. Tonight's talk will be on avian locomotion and parrot skulls. Our speaker tonight is Dr. Armita Manafzadeh. She's currently a National Science Foundation postdoctoral fellow in biology and Gaylord Donnelley postdoctoral environmental fellow at Yale. She earned her BA in Integrative Biology from Berkeley in 2016, and her PhD in Ecology, Evolution, and Organismal Biology from Brown in 2022. Armita is interested in how joints like hips, knees, and ankles work and where they come from, both evolutionarily and developmentally. And she's also going to fit in a little talk about parrot skulls. And maybe we can why parrot bites hurt so much. So with that, I will turn it over to you, Armita.
Fantastic. Let's see if I can get Zoom to cooperate. Thanks so much, Amy. Can you all see that slide? Yes, fantastic. So as Amy said, I'm currently a postdoc at Yale. I thought I'd start by giving you a little bit of context for kind of what my research is overall, and then I'll focus in on our bird joint work that we're going to talk about today. But this is really fun. It's such a cool idea that you all have this group, and I'm really excited to talk to you all. My work in general focuses on joints across all vertebrate animals, so animals with backbones. And the reason I study joints is because they underlie this really incredible diversity of vertebrate animal motion. So all the stuff you're seeing on this slide, things from birds moving and doing mating dances and so on, and feeding and locomotion across all of these different animals, but also all of the other stuff that animals just do with their joints, like grooming and hanging out. And I think studying joints is really important because they underlie this animal motion. So if we want to understand how it changes over developmental time, as these organisms grow from juveniles to adults, or over evolutionary time across some of these major transformations in vertebrate animal history, life started out in the ocean, vertebrate life, and then came onto land. Or if we want to understand how animals are adapted to all kinds of different habitats, or maybe their potential to adapt to new habitats as climate continues to change, then understanding how their joints work is a really important part of that process. And you might be a little familiar with your own joints, a little too familiar sometimes if you've had some pain. So you know that joints have all these different pieces, things like cartilage, things like the ligaments and capsules that connect bone to bone, the bone itself. So my research really focuses on answering two questions, and that's how do joints work at a really basic level? How does the shape of joints relate to their function, to the motion we see them using? And also, where do they come from, developmentally and evolutionarily? So I look at a whole lot of different joints from across that vertebrate tree in all kinds of animals. And these span a whole range of different animals, including those parrots, which I promise we'll get to at the end of the talk here for a little bit. But parrots are actually in the minority of the birds that I look at. So most of my time is spent looking at helmeted guinea fowl. And if any of you have a backyard flock or have a friend who does, you might have seen some of these guinea fowl. They're birds that are endemic to Africa. They look kind of like polka dotted chickens, but they're great backyard flock birds because they love to eat ticks. And they're great research birds because they're really good at walking on treadmills. So we love to study our guinea fowl. So today, I thought I'd split my presentation for you all into three parts. So I'll talk about two main projects that I've done looking at the evolution of avian locomotion. And then that third part will be a little teaser of some parrot work. It's work that I'm a collaborator on. The primary author is actually another postdoctoral scholar down at Duke University. But I highly encourage you all to invite her to do another program for you all if you'd like to hear more about that work when we get there, because she's done a ton of this parrot work and she's really great. For the work that I've led, I thought we'd start by talking about paleontological reconstructions, which I can see I have a typo in that slide already, which is a great start. I promise there won't be too many more of those. But I'm situated here at the Yale Peabody Museum, so I've done a lot of work on paleontological reconstruction. And I think it's really interesting to think about how paleontologists work with fossil bones and how they take those bones and try to understand how extinct animals like dinosaurs would have moved when they were alive. So if you think about the data a paleontologist has to work with when they're trying to do these reconstructions, they're picking up fossil bones, and I've got some bones right here. They're holding them in their hands, and they're trying to get a sense of how those bones fit together and what poses seem reasonable at a joint and which ones seem impossible at a joint. As they're doing that, there are some basic intuition elements that go into that. There are some data elements that go into that. But for centuries, from kind of the 1800s, back when dinosaurs were named, till now, they're basically using protractors to measure how much motion they think is possible at a fossil joint. And they're reporting those excursions that they think are possible, what rotations they think are possible, on bar graphs. This is truly, and I say to you, you could walk into a museum collection, you could all do this yourselves. They're picking up these bones, they're manipulating them in their hands, and they're using a protractor to measure those angles. So this is kind of the field standard way of approaching reconstructions of extinct animals like dinosaurs for something like 200 years. The issue with this is if we consider a joint like a bird hip joint, we could do something like that. We could decide how much we think it flexes and extends, how much the thigh bone moves forwards and backwards. But it takes just a couple of seconds of watching a real bird walk around to realize this is a totally three-dimensional problem, right? Our animals aren't actually just moving in a single axis. So those protractors aren't really going to cut it and help us understand how their joints are working and how they were moving around. So when I got into this field, I got into this from a paleontological perspective. I did my undergrad, as Amy said, at UC Berkeley, where there's a really great paleontology museum as well. I was looking at fossils and I was trying to figure out how to reconstruct them, but I realized this single axis protractor approach wasn't really going to cut it. So what I did instead is I took inspiration from computer animation. If you've ever seen a Pixar movie, or your kids or grandkids who've ever seen a Pixar movie, they animate the characters in those movies. Like we have Sully the Monster from Monsters, Inc. on the slide here by using virtual skeletons. And the skeletons that Pixar uses to animate these characters are made up of virtual bones strung together by virtual character animation joints. That's a really cool technology for Pixar and these animation studios to be able to use because it helps them create this lifelike motion on their characters. But we can steal their methodology and we can steal their software and we can use that to do science. And that's exactly what I do in my research. So I take those character animation joints and I assign them to real anatomical joints from birds and other animals. Here we have one of those guinea fowl hip joints. And once I create that framework on my computer screen, I can then apply combinations of rotations and motions to those joints in a systematic and reproducible way. So instead of just moving the bones in my hands in the museum, I can move models of those bones on my computer screen using these animation joints to get a sense of how they might be able to move. And then for any given configuration of those bones and how they fit together, I can get the computer to tell me, hey, those bones are inside of each other. That's actually not possible. So I can exclude the impossible poses from a pair of bones and help use that to reconstruct the motion for any given extinct animal. Now that's a big advance because it helps us move out of a single axis system. We can consider complex motions at these joints, the way we know that birds actually do move, dinosaurs actually do move, but it only takes into account whether the bones are inside of each other. And if we think back to how paleontologists work with fossil bones when they're trying to figure out what poses these extinct animals used, they're not just asking whether the bones are banging into each other. They also have that intuition for whether some joint poses look right or look wrong. And that's why they're reconstructing their extinct animals a specific way. So if we think about how people have done that, they haven't really formalized that intuition super well. I'd say more often than not, they've just truly written things in scientific publications like, this seems correct or this seems incorrect, which is not very helpful. But if we try to get a sense of why they think things look incorrect or correct, here we have the neck region of a long-necked sauropod dinosaur, kind of like the Brontosaurus. It's the centerpiece of the Peabody Museum if you visited us here at Yale. And if we look into the joints of that neck region, they've done things like look at the regions of the bones that overlap with each other at a joint, and they've set criteria like maybe those bones need to overlap by 25% or 50% or 100% for that to be a viable joint pose. The issue is that we know that overlap can't be the full story when we think about these joints, because if we look at these knee poses along the bottom of the slide here, all of these knee poses overlap by the exact same amount, but some of them look right to our eye, that's the green checks, and some of them look wrong to our eye, that's the red X's. So I got to thinking that we could improve our reconstructions of extinct animals and our understanding of how joints work in general if we could better formalize this intuition and figure out why things look right or look wrong to us and how to describe that. So to do that, I started by looking at bird ankles. Now, you're all bird enthusiasts, so you know that birds do this very silly thing where they fuse up the sole bones of their feet into a single bone. And so their ankle joints are these simple hinges that people always think are backwards knees, but we know they're not. And so if we take an ankle joint from our guinea fowl and we zoom in on that ankle joint, we can focus in on the regions of the bone that actually interact with each other, the articular surfaces that form the joint in that ankle. And we can use that same animation software that we use to do a lot of my research to send a series of little arrows from one joint surface to the other in 3 dimensions. And when we send these arrows across our joint surface, they can help us understand how those joint surfaces are actually interacting in 3 dimensions. So we can do things like measure how long the little arrows are, we can measure what angles they're coming off at, and that helps us get a quantitative number-based understanding of how these bones are fitting together inside a joint. So if we look at this guinea fowl ankle configuration that I have on the slide, that looks pretty good to us. I've colored it Goldilocks yellow because we're pretty happy with it. They're pretty intermediately far apart and we're okay with that. It turns out it's a lot easier for us to identify when joint poses look wrong and to describe why they look wrong. So if we take our guinea fowl ankle and we slide those joint surfaces apart, that's a situation that looks wrong to us in low overlap. And we've always known that that's a problem. We can't have that at a joint. But even if we maintain overlap and we move the 2 halves of that ankle different distances apart, that low symmetry situation also looks wrong to us. And even if we maintain overlap and symmetry, but our joint surfaces hit each other at a kind of funky angle, that low congruence situation looks wrong to us too. So we can take these 3 criteria, low overlap and low symmetry and low angular congruence, and those kind of capture our full intuition for why certain joint poses look wrong to us. The great thing is, because we've done this in our Pixar animation software environment, and we've used these tiny little arrows to connect our bone surfaces so we can measure things like how long they are and what angles they're at, we can use all that data to help us come up with a joint-specific articular score, a score that explains to us how well or how poorly the joint surfaces fit together from 0 to 100. So instead of just saying this pose looks right or this pose looks wrong, across a ton of different ankle poses, anything you can dream up in your head for these 2 guinea fowl bones, we can give them a score and say, well, this one's really, really bad and this one's really, really good. And now we have this whole spectrum of different poses and numbers and scores that we can look at. So the way we go about doing this for any single joint, this can give us a sense of which poses look right and which poses look wrong. So we could start with a theoretical space. We don't have to get into the math of all this. It doesn't matter if you don't like numbers. Every little blue dot that you're seeing on the screen is, in some measurement system, a different pose that a joint could be in. All right, every single dot is a different pose a joint could theoretically use. What we've got on the right-hand side of the slide is a guinea fowl ankle joint rotating through all those different poses that it could maybe potentially use. At every single one of those poses we can spin to, we can stop our joint, and we can say, all right, you've rotated to a certain pose. Now, why don't you slide through all the different ways that you can slide as surfaces? So you're not rotating, you're just translating, you're sliding. And at each one of those, we can use this little system we've come up with to come up with an articulation score for how well the bones fit together. So some of them are going to be really good, some of them are going to be really bad. We can do that across this whole theoretical space of potential joint poses. That gives us something like 7 million joint configurations to score. So if you're a paleontologist, there's absolutely no way you could sit there and do every single one of these by hand and consider it. But because we're able to take advantage of this new computer software that I've developed, we can do this automatically on a computer screen. So I set up my analysis, I go to bed for the night, I wake up in the morning, and I have magical results on my screen, which is really convenient. So if we do this for our guinea fowl ankle joint, let's say that that's our dinosaur, right? Let's pretend we didn't know anything about how a guinea fowl moved. If we start with its ankle joint and we had all of these possible poses that we thought maybe it could use, we can rule out the poses where the bones would bang into each other. And if we rule those out, we're still left with a ton of possible poses, but we've gotten rid of a lot of that space, right? These are all viable. They seem like they could be possible. But we can now go a step further and we can take those articulation scores we've come up with. We can map those onto this distribution and color the points by how high or low their scores are. And now instead of just this binary possible or impossible, where we have to exclude some poses and allow others, we can get a sense of how the ways the bones fit together changes across potential pose space. And then we can use those scores to narrow things down to look at only higher and higher scoring regions of pose space. So if we do that for a guinea fowl ankle, the highest scoring region of all the possible joint poses is this really, really tiny region. And that's interesting. It tells us that there's some specific poses where the bones fit together especially well. But that just tells us about how the bones fit together. It doesn't tell us anything about what birds actually do with their joints. So to understand what birds actually do with their joints, we need to take these data from my computer screen and compare them to how birds are actually moving in life. To do that, we could look at our guinea fowl walking on a treadmill in the lab. And this is what I did a ton of during my PhD research. But the problem here, right, if I'm interested in hind limb joints, sometimes we can see the ankle. Sometimes it's hidden up there underneath all the other feathers. And the other joints of this bird are hidden up underneath the feathers too. And that's no good because we want to be able to measure their motions in a ton of detail to really understand what's happening at the bone-bone interaction level. So the way I get around this is I use a method called X-ray reconstruction of moving morphology, or XROM. This is a super cool technology. The idea here is instead of just taking a video of our bird walking on a treadmill, we still have our bird walking on a treadmill, but we take two high-speed X-ray videos of that bird walking at the same time. So instead of just a single static X-ray like you might get at the dentist or the doctor, we're taking a whole video in that X-ray view, and we're taking two of them at the same time. In each of those views, we can track the positions of specific marked points within the bird's bones. So we can get a sense of how's its thigh bone moving and its shin bone and its foot bones. And we can see how all that's moving in these 2D spaces. And then we use a computer screen to recalibrate where these two X-ray cameras were as they were placed in the lab in 3 dimensions. And we take CT scans of all the bones of our birds so we can get 3D models of them. And we use the data we've gathered to reanimate how the bird's bones were actually moving inside its skeleton as it was alive in our lab. So we're getting this 4D X-ray vision into the skeleton of the animal, and we can have it doing all these different behaviors and figure out how its bones are moving in super high detail. When we get these XROM derived data, these data about how the bones are actually moving while the bird is moving around our lab space, we can compare them to those articulation score data we got. And what we find is that the poses the guinea fowl ankle actually uses while it's walking on our treadmill fall within that highest scoring region of pose space. So those high scoring poses are red, and the black dots are tracing out the poses that the ankle is actually using in real life. And that's not just a one-off result. We have multiple different individuals of guinea fowl walking on treadmills at all kinds of different speeds, walking and running. And all of those black dots represent poses that the guinea fowl actually used in life in the lab, and those are all falling in that highest scoring region of pose space. So what we've been able to do is come up with a way to start with the shapes of just the bones of the skeleton of our bird and use those to predict how this animal is actually walking. And that's a really cool connection if you think about paleontology, because it suggests the shapes of the bones can tell us how animals actually move when they're alive. Now, we thought this might be something weird about the guinea fowl ankle, and that wouldn't be great. So we also looked at the toe joints in the guinea fowl, and we found that same connection. The highest scoring poses in the system are the ones they're using when they're walking around. We did this for emus. We took videos of emus walking. They're from a different part of the bird family tree. And we got the same results for their ankles and toe joints, all of which have this bicondylar two-halved geometry. We also looked at moas, these extinct giant ground birds, kind of like terror birds, right? These big birds that live down in New Zealand and other places. And when we looked at how their bones fit together, that matched up with our predictions from the scientific literature of how people think these birds move. So all of this gave us some confidence that we found a connection between bird joint shape and how these animals actually move in life. So we went a bit further afield in the bird tree of life. And if you've ever driven down I-95, you know where we're headed with this. That big fuel tank sitting there on the highway tells you that Deinonychus, this dinosaur, is our New Haven treasure here at the Peabody Museum. So if you haven't been to Yale campus, I'll tell you that Deinonychus is our raptor, our dinosaur here. It was discovered by Yale paleontologist John Ostrom back in the 60s. And we love Deinonychus here because it was a key player in our understanding that birds are living dinosaurs and really piecing together that connection in the 60s. It helped us understand how and why birds have such high metabolism, how that came from some of their dinosaurian ancestors, and how deep that goes back in history. This is a cast of Deinonychus, this dinosaur that's hanging in the stairwell here in one of the buildings at Yale. So if you're ever on campus, pop into the Environmental Sciences Center, which is the building I'm actually sitting in right now, and check this out. It's open to the public during the day. even if you don't really care about dinosaurs at that level, you might recognize this because it's your Jurassic Park raptor. So we now know that these are feathered animals, which is not how Jurassic Park portrays them. But Deinonychus is a respectably sized, scary raptor, and actual velociraptors are kind of tiny and turkey-sized. So the creators of Jurassic Park reached out to Yale paleontologists. They got information about Deinonychus, and this is the raptor in those movies. Now, what's fun about Deinonychus, we have these original fossils here at Yale. And so as part of my postdoc, I was able to pop downstairs to the museum collections and CT scan the bones that make up the foot of this animal. Now, Deinonychus is named Deinonychus because that means terrible claw. And I think you can see which claw we're talking about there. I would not want to get on the wrong side of that thing. But what's cool for applying my joint-focused methods to this is if we break up the foot of Deinonychus, it has 12 of those different bicondylar simple joints that make up its foot, and they all have different geometries. So if we're able to use this kind of articulation score approach, we should be able to look at those 12 joints, get different results for what poses those joints should use, and help us reconstruct how Deinonychus would have walked when it was alive. So we did this. We have 12 different graphs here for all 12 of those different joints. We can focus in on the highest scoring regions of joint pose space for each of those 12 joints. And by using the constraints that we've learned from guinea fowl and other modern birds, we can reconstruct a data-driven stride cycle for this dinosaur. So we can take the fossils out of our museum drawers and reanimate them using these data to understand how the dinosaur would have moved when it was alive. Now, what's fun about being here as a postdoc at Yale is I got to hand off these data to professional animators. They made that animation that you're seeing in the upper left of a nice fully feathered Deinonychus walking around, and the feet of that stride cycle are directly inspired by our actual scientific data about how the foot of this animal would have moved. And what you're seeing in the bottom of the slide is a tiny little snippet of the renovated museum. So there is a Deinonychus on display right there, and there's a cute little touchscreen kiosk next to that, where this animation's on display, and you can see the evolution of locomotion from dinosaurs on the bird line to birds, seeing how those transformations have happened. So it's a really cool part of the renovation. I encourage you all to check it out. For those of you who popped in a little bit late or maybe watching the recording, the Peabody Museum at Yale is now fully free to the public for the future in perpetuity. So please do come by and check it out. Bring your friends and family. We can also use these data to look in at that terrible claw on the second digit of this animal. So this claw, obviously, it's big, it's exciting, it's drawn a lot of attention over the past decades. And when we look at it, it's been implicated in all these different behaviors. People want to know, is Deinonychus stabbing its prey or slashing its prey or jumping on top of its prey? It's dinosaur paleontology, so everyone wants to talk about how these animals are attacking each other. Well, now we can actually answer these kinds of questions with real quantitative data, right? So instead of just saying, maybe it could stab or slash or whatever, we can formalize these hypotheses of what its joints would have to be doing to enable that. So if we focus in on this joint, we can say, well, if it were slashing, it would have to go through an arc of this many degrees. The score for the pose at that joint drops to a certain amount, and maybe that's lower than what we're seeing in any of our living animals that we've studied. So we can confidently rule out a slashing behavior. So we think that Deinonychus was probably stabbing with its claws. What's really cool is we can look at fossils of closely related animals that lived in Mongolia and different parts of Asia. And we find some of these dinosaurs preserved with their claws stabbed into the necks of their prey. And so we see direct evidence that this is probably what they were doing with their claws. So this is our first little vignette, our first story. That's on the dinosaur side. I want to give you another story about the evolution of the avian knee. And this is that drumstick story that some of you saw sent around before this talk. But I want to break it down for you because it's stuff that you can see at your kitchen table. And I think some of that might be fun for you to understand the anatomy that you're looking at, even in things that you might be cooking or eating for dinner. So if you've ever eaten a drumstick, you're looking at the shin of the bird, right? Many of you are probably familiar with this. But that shin region of the bird, the drumstick, is made up of two bones. There's a big dominant tibiotarsus or tibial bone, and that's what sticks out of the end of the drumstick. There's a second smaller bone called the fibula, and that's also hidden under the meat there. So I've gotten some blown up guinea fowl bones here. If you can look at the speaker view, you can see how tiny and kind of toothpick-like that fibula is compared to the big tibiotarsus. So there's a big contrast there. And when we look at these bones, that's a very strange condition that we see across birds with some variation, but roughly across all birds, we're seeing something like this. But if we compare birds to their archosaurian relatives, the crocodiles, and a bit further in the vertebrate tree of life, we also look at lizards, the reptilian relatives. That's a really unique configuration that we're seeing in birds for two reasons. One is that in birds, the fibula is reduced. It doesn't reach down to the ankle joint. So in lizards and in crocs, it really is reaching all the way down to the ankle joint there. The second is that in birds, there's this mid-shaft articulation, this joint between the fibula and the tibiotarsus. And you can see that in the crocodile and the lizard, that's not the case. Those bones are far apart and they maintain their distance along their lengths. So that's a unique configuration in birds. And some folks have thought about that for quite some time. So developmental biologists who work with embryonic chickens have really tried to understand how the fibula gets to be so short, when in development it gets to be so short, why it's so short. So they've looked into that and studied that in various ways. But people have thought for a long time that this reduction of the fibula, the shortening, can't have any kind of a functional significance for the locomotion of these animals. And I thought that was a little suspicious, and I'll explain to you why I thought that was a little suspicious. If we think about traditional reptile locomotion, you think about an iguana or an alligator moving around, they're typically using this sprawling posture with their limbs out to the side. It's a pretty characteristic posture, and they're walking around on all fours. And to achieve that traditional picture we have in our heads of what it means for a reptile to move around, if you look at the back end of an alligator here, at their hips and at their knees and at their ankles, there's a ton of motion possible at each of those joints. They're all moving around a whole lot. And the way they're doing that is through this linkage system that connects the knee to the ankle. So because the tibia and the fibula both extend from the knee joint all the way down to the ankle joint, there's kind of a little 4-bar linkage system that connects up those bones at the joints at the top and the bottom. And that little rectangle of sorts kind of tweaks into a parallelogram as these animals move, but the motions of the knee and the ankle are coupled to some extent. And we look at real data from an alligator moving in the laboratory, one of those X-ROM animations, we can see that's essentially the case. The tibia and the fibula are kind of moving back and forth in a linked way. Now, this works perfectly well for an alligator, and they're using that 4-bar linkage. But if we look at the lizard and the alligator, that mechanism, which is called the curl mechanism, which is just a fancy word for what we just saw, is used by both of those animals. Now, the problem is, and the reason I think that fibular reduction is probably going to be important for birds, is if you look at the bird lineage, they lose the ability to move their hind limbs, their legs in that way. Let's look at why I say they're losing that ability. So if we think about an alligator skeleton versus a bird skeleton, there are a ton of differences in how the entire hind limb, the leg, is constructed. So these are the fancy names we give to all the bones in the limb, but let's focus in on some of these differences that we're seeing in the limb skeletons between an alligator and a bird. One big one is in an alligator, you have the tibia, and you have the proximal tarsals, which are some of the ankle bones in the alligator. Those will get fused up into a tibiotarsus in the bird. So that's why it's called a tibiotarsus. It's the tibia plus the tarsals glued together skeletally into a single bone that makes up most of the shin. So that's one big difference. Another big difference, which we touched on previously, is they're taking all those metatarsals and tarsals, these sole bones of the feet, and birds are fusing those up into a single bone called the tarsometatarsus. So that's a big difference. It's something that birds are doing that's special. And if we look at that fibula, we've talked about how the alligator has this big, long, tubular, robust, big fibula, and birds are reducing that into this split-like tiny toothpick structure. So when we look at the full limb, I think all of those changes together mean that that little linkage system we had in the alligator isn't possible in the bird. It's broken because of that fibular reduction. And what that means is that this highly mobile ankle that we saw in the alligator gets reduced into being a simple hinge in the bird, which is what we saw in our last big study looking at guinea fowl ankles. But it also means that people have assumed that this highly mobile knee joint present in the alligator gets reduced to being a hinge joint in birds. It's traditionally thought that birds are hinge-like knees like ours. And, you know, that might make sense. If you watch a bird walking on a treadmill, it seems like it's moving kind of in one plane. It seems like its knees might be hinges. And it's very common across animal biomechanics for us to assume that the joints of other animals move a lot like how our joints move, even when that's not fair. The issue is that if you watch a guinea fowl moving around in the lab, it's spinning a whole lot, right? And I'm sure your parrots are doing the same thing. They can move in all kinds of crazy ways. They're not just moving in straight lines. So that means that we have to have an ability to break out of the plane coming from somewhere. We can't just have a series of hinges. Where we see this happening is happening at the knee joint in birds. So we've used these extrom techniques to look in at the knee joint, and the knees are undergoing a motion called long axis rotation, where the entire shin, the entire drumstick of that bird is spinning on the thigh bone by over 100 degrees. So it's really painful for us to consider. I wouldn't want my knees to do that, but birds are doing that routinely across all kinds of different species of birds. And what this does is it serves to reorient the foot so they can break out of that plane. And when they're maneuvering in 3D, they're using this knee long axis rotation to do that. So that got me thinking that there might be some connection between the reduced fibula and that change of hind limb architecture in the skeleton of birds and the capacity of birds to use this extreme long axis rotation potential. So how did we get at this? We took our 3 different living reptiles, the iguana and the alligator and a guinea fowl, and we took videos of them moving around in the laboratory. We've talked about how standard videos don't really let us study the skeleton in a ton of detail. So we also took X-ray videos of these animals moving around. And those X-ray videos let us use this X-rom technology to get 3D animations of all of these animals moving around the lab space. Now, when we did that, we could focus in on how the drumsticks of these different animals were moving. So if we do that, we can focus in on the tibia in gray versus the fibula in blue. And if we focus on the extremes of those motions, we see something really interesting, which is that in the iguana and the alligator, these animals that I said are kind of traditional sprawling reptiles, they're sharing this skewing pattern where the fibula kind of rocks back and forth relative to the tibia across different behaviors. But the bird is doing something else entirely. In the bird, we see this unique rolling pattern of the fibula where it's spinning about its long axis and kind of hinging off of the tibia. And it seems to be hinging off of the tibia at the site of that unique joint that the 2 bones have along their length. And where we see this happening most prominently is as our birds are using that knee long axis rotation motion and maneuvering and spinning in 3D in the lab. So it seems like the reduced fibula and this complete transformation in the hind limb skeleton in birds is enabling these birds to move around the world in 3D in the way that they do. So to get a sense of how the reduced fibula is really enabling this extreme motion, I focused in on the knee joint and watched it spin in that long axis rotation behavior. And we can use our X-rom animations to focus in on the joint surfaces. So if you look in at a bird knee joint, we're looking in from the back of the animal, we can put those little rays, those little arrows that we used earlier, back onto the joint, and that helps us study how the bones are interacting with each other in more detail. When we do that, we can watch the joint as it spins throughout those long axis rotation sequences of over 100 degrees of rotation. And we can break the joint back open and look inside of it virtually on the computer screen. Our bird is just fine, I assure you. And we can see what joint regions are being used. And if we do this across an entire sequence, as the shin spins outwards, we can see how those interactions change and which regions of the bones are involved. And by putting all these data together, we can get a sense of how the different regions of the tibia and the fibula and the femur, the thigh bone, all interact with each other. So what we find is that the bird knee joint, and this is pretty conserved across different bird species, consists of 3 main articulations. So on the medial side, on the inside of the joint, we've got this interaction where we can put those rays back on to see it in more detail. We've got this arc-like surface, which is really setting up an arc for the thigh bone to rotate on. So that shape of the joint surface seems really important. On the lateral side of the tibia, we've got this conical surface, and that conical arc-like surface also seems to be setting up an arc for the thigh bone to rotate on. And what's arguably the most interesting is over on the lateral side of this joint, towards the outsides of the animal, we've got this fibular articulation, where there's a special shape of the thigh bone that holds the fibula in place. That fibula isn't really moving relative to the thigh bone very much at all. And what that means is that the fibula is kind of left behind as the rest of the thigh bone, as the rest of the tibia, the rest of the drumstick continues to rotate away from it. So we've got this extreme rotation between the tibia tarsus and the fibula. And this is what's enabling that extreme long axis rotation in birds, because the reduced fibula gives it the freedom to move independently. So once we understand this configuration really well in birds, and that's what we've done here, we can then do the same thing for alligators, which I won't take you through because this is the parrot club, so we care most about the birds. But I'll assure you that once we do this, we can see how these different features, like that arc-like tibial surface and the reduced fibula and the connection between the bones, are all working together to allow this bird's knee joint and complete hind limb to work in a completely different way from its reptilian relatives. So this is where we really need the evolutionary perspective. And if we create a family tree of vertebrates, if we didn't have any fossils to look at, if we could only work with our living animals and the data we have, we might think, well, alligators are retaining from their ancestors a capacity to rotate their knees quite a bit. And somewhere on the line to birds in evolutionary history, they've slowly and steadily increased the capacity to rotate their knee joints. And we see this in modern birds today. But when we actually look at our fossils in the museum and we get some dinosaurs into here, we see a completely different story. So what we actually end up finding by looking at these fossils is that there's a stage where knee rotation potential decreases extremely. So in big meat-eating dinosaurs like Allosaurus, which is really similar to a T-Rex, you can picture that, we get this condition where the tibia gets an extra piece of bone that reaches across to the fibula. That's indicated by that little black triangle there. And that skeletally glues together the drumstick of these big dinosaurs. If you look at that yellow tibial surface, it also becomes straightened out front to back. And these things put together means that the knee joints of these big meat-eating dinosaurs are essentially locked into being hinge joints. So this is where the real hinge joint is happening. And then at some point in evolutionary history, between these big meat-eating dinosaurs and the birds we have alive today, there's this extreme mobilization of the knee joint. And that's enabled because the fibula is released from the ankle when it reduces. And that gives it the capacity to adopt that new rolling motion pattern that we see. And that, coupled with the increased curvature of those tibial surfaces, means now we have all the ingredients we need for this bird to be able to have this extreme knee long axis rotation. So like I said, we see that extreme knee long axis rotation enabling tons and tons of maneuvering and motion in the lab. We see this in our guinea fowl, but we also think, and this is completely understudied, and so maybe this will help you see your parrots moving around in a new light, but we think it's affecting a ton of different bird species and their ability to move around their environments. So things like loons with their hind limb propelled diving, various kinds of birds moving around on different substrates, on grass and in different environments, fast cursorial birds that are running around and moving at different speeds. We really think that the modern avian radiation, all of these birds that have adapted to all of these different lifestyles, are capitalizing on this ability to rotate their knees in really extreme ways and using it to adapt to their conditions of life. So if any of you happen to see your parrots doing crazy things with their legs, and now you know that their knees are capable of doing this, I'd be really curious to hear from you about what you think parrots might be using this for. As a final little bit, and this is only going to be a little piece here, I just want to show you some parrot skulls because I know you're all going to like this, and this might help you see those in a new way too. This is work led by my friend and colleague, Dr. Melody Young, who's currently a postdoc at Duke University. Please do invite her to give a seminar if you're interested in this work because she'd be awesome. She's done a whole lot of work with lovebirds, and it's really incredible. But I'll give you a taste of some of it that I helped with. Melody's really interested in this idea of how parrots use their skulls and use their beaks to assist in their locomotion. So if you have a lovebird and you give it different inclines to walk up, if you just have it walking on flat ground, it'll of course walk across the ground. But as you increase that incline, they'll start to use their tails to help them move. They'll get the tails in there. And then if you really start to make things steep, they'll actually use their beaks to help them climb. And so you're seeing different percentages of the time that these animals are using their tails and their beaks to help them climb. What's exciting is if you put these birds on a vertical incline, and I just love watching these videos of the lovebirds doing this, you can see them grabbing on with their beaks and using that to help pull themselves up surfaces. And you really only get this when they're on these vertical or close to vertical inclines. So the graph on the right is showing you the left and right hind limbs being used in the blue bars. The orange bar is when the beak is being used, and the green bar is when the tail is being used. And these are different sequences in the progression of this bird climbing up. So you can see there are some stages, and I'll see if I can get my mouse on there. Maybe you can see where all 4 of those structures are being used at the same time for this animal to move up, but there's different timing of all of those pieces working together to help this bird move up the incline. So this is something Melody had figured out. And when I worked with her, she said, let's look at some lovebirds and let's figure out how they're using their beaks to help them climb. Let's like get into the joint in the skull. So she knew parrot locomotion really well. I knew joints. We got together and teamed up to try to figure this out. And we CT scanned some lovebirds and we looked into their skulls. And when we did this, we were able to virtually take slices through their skull shapes and see how the joints in their skull work. So there's this really cool joint between the upper beak and the rest of the skull that has a ton of mobility in these birds. And this is something that's true across a ton of birds, but this capacity to move the beak relative to the rest of the skull is really extreme in parrots and in lovebirds. So many of you likely know this is the rotation potential of a parrot skull. I believe this is a macaw skull from our colleague Larry Whitmer, but you can see how much these bones have the capacity to move relative to each other within the skull. This is really weird. If you think about your own skull, your skull bones definitely can't move relative to each other that way, but birds do this all the time. And this cranial kinesis behavior is really, really elaborate in birds like macaws and lovebirds. So because of this hinging of the upper bill relative to the rest of the skull, we wanted to see whether they could actually use that motion to generate force to help them climb, and if that's what's going on. So Melody and her colleagues built this really cool little bite force measure for parrots. So it's a custom 3D printed bite force measure, and they got their birds to clamp down on it. So if you've been bitten by your parrot, you know they can bite really hard. So these parrots love to bite down and snap, and they can generate a respectable amount of force with their little snaps. So we were able to measure how much force they could generate using this really cool contraption. And then we got to see, this is a complicated graph, we don't really have to care about it, but as these parrots are climbing up the wall using their beaks, what we found is that they're actually able to generate force. And in the same way that they can bite down and use that behavior when they're eating, they can also generate force to propel themselves and pull themselves up inclines. So they're using different patterns of that beak versus skull motion when they're biting down on food or biting down on you versus when they're moving up a wall. So this is a really cool elaboration of this cranial kinesis mechanism that we know that all birds can use. And parrots really seem to be doing something special where they've co-opted that. They found a way to use that to help in their locomotion as well. So I think this is a really cool story of how studying the anatomy of the joints in the skull in these parrots is helping us understand this locomotor behavior and how they're taking advantage of something that is ancestral to birds, but using it in a new way to help in their locomotion. So that's just a tiny taste of that. I think it's really exciting stuff. I'm sure Melody would love to talk to you all if you want to hear more about it, but thanks so much for inviting me to chat, and I'd love to answer any questions you all have about any of this work.
Wow, that was fantastic. So fascinating.
Thank you.
Basically, the 2 take-homes are we have really crappy knees compared to
Mhm.
other animals, which probably explains why our knees break down so easily in humans. And second, parrot beaks are basically ice axes that are attached to their faces.
They can bite hard. I'm
Yeah,
sure you've experienced that.
I'm not picturing, when you climb an ice fall with ice
Yeah,
axes,
just grab on there.
well, that's what they're doing with their beaks.
Totally, totally.
I actually always felt like, looking at my birds, it always reminds me of the ferry. Like, if you ever taken the ferry to Long Island, the whole front just lifts up. To me, it looks like a parrot beak. So when my parrots lift just their top beak up, it always looks to me like the ferry.
I love that. I'm definitely going to use that in talks from now on because you're right. It looks exactly like that. And people have a really hard time. I mean, it's great that you've seen that motion because I think people who don't spend time around birds are totally confused. They're like, what? My nose can lift off the rest of my head. They have absolutely no idea what to picture, but you can see it so clearly in a bird.
I mean, I've had parrots for decades, and I never get tired of watching that beak just move by itself. It's
Oh,
just
it's ridiculous. It's the coolest thing.
so strange. And knees, I honestly did not know until this talk that parrot knees rotated. I had
Mhm.
no idea. Now I'm gonna have to like go back and watch my birds because I I just never noticed it. Now I've got to see if I mean, that would be incredible if our knees were able to rotate like that. I mean,
Totally.
think
Yeah,
of
we just have a completely different setup.
it's unbelievable. So Rebecca says, really interesting work. How do you determine how much soft tissue to calculate for in extinct species joints? Good.
That's such a great question.
Cartilage, yeah.
That's something we think about a ton. So yeah, for those of you who might not be as familiar, if you dig up fossils out of the ground, right, they're going to look like separate bones. Bony tissue fossilizes really well. It infills with minerals and we can get those hard tissues preserved. But soft tissues, things like cartilage, things like ligaments, muscles, tendons, we don't typically get those preserved in the fossil record. Sometimes we do in really exceptional cases, but for the most part, that's all gone. Which raises the question, if you want to reconstruct the function or the motion or the locomotion of these extinct animals, how do you work with these limited data, just bones? So it's really cool. The first study I showed you, where we reconstructed the guinea fowl and then Deinonychus, all of that was done with no assumptions about soft tissues at all. So that was just working with bones without any assumptions about soft tissue. That's exciting for us because it means we don't have to make assumptions about soft tissue. We don't need to make up how much soft tissue we think was there. There's a signal preserved in just the bony tissue that tells us about locomotion. Now, what's exciting about birds and closely related dinosaurs, so things on that meat-eating line, is that they have really thin cartilage. Humans also have quite thin cartilage. Other animals in the vertebrate tree, things like alligators, have really, really thick cartilage. And so it might be harder to understand how animals with thick cartilage might have moved based on just their bones. But for things with thin cartilage, we can get a really good sense from their bones. Now, sometimes that's not enough, like I mentioned. So some of the work that I'm doing to help us get around that issue is to try to reconstruct soft tissue onto our fossils and use that to constrain our animations of their motion. And the really cool method that we've been using to do that is, if you've ever seen one of those Pixar movies, if you imagine animating a character on a screen, one of the hardest things these animation artists are confronted with is how to animate clothing, right? Because you need something that behaves like cloth. You need to move and slide over your character, and you can't have it go inside the character. It has to like glide over their motion gently. So we're able to use those same simulation methods that they use to simulate cloth, like tablecloths or clothing or flags. And we use that to create virtual ligaments, those tissues that connect bone to bone. And because those simulation methods are created to allow something like a tablecloth to fall over a table, our ligaments can also slide over bones without going inside of them or behaving in strange ways. So we're really trying to adopt a lot of these methods across different fields to help us work with this kind of research. So we're getting better and better at simulating those soft tissues. And that's something we're adding on to our reconstructions all the time.
That's incredible. Do you ever need or want to access any proprietary information from places like Pixar? And have you ever collaborated with them?
We haven't collaborated with them directly. So there's a software that's very similar to what they use that's actually completely free to educational users. It's called Autodesk Maya. So it's been really incredible for us because we're not using it in a commercial way. We're just academics trying to do research. We've had totally free access to it, along with those really awesome algorithms that have physics-based simulations of how things like cloth deform. So we've been able to access a lot of that completely for free, which is really incredible.
So Dawn asks, do you do any dissection to look at tendons, et cetera, in living birds?
Yes, absolutely. So that work I mentioned, I haven't gotten into tendons as much because tendons and muscles are a whole different world. So if you think about how tendons and muscles work, sometimes they're active, sometimes they're not active, and they activate in all of these really complicated patterns that help birds and us and every animal move. So because we can't figure out what those activation patterns would have looked like necessarily for extinct animals. I don't do that kind of work. There are people who've done really exciting research trying to simulate those patterns, but I haven't gone there. But for the ligaments, I've spent a ton of time dissecting birds, trying to get a sense of where those ligaments attach bone to bone and how variable that is across different species, because we need to understand how variable those attachment sites are to understand how to triangulate that back to the fossil record and reconstruct that onto dinosaurs and things like that. And what's interesting when you do those dissections is you find there are some things that are described really well in the scientific literature and other things where in 1700 whatever, there's some guy in Germany who said that this is what the structure looks like. And then everyone for the next 200 to 300 years said that guy in Germany said so, so that must be the way it is. And then you look at an actual bird and you're like, that's not the way it is at all. Just he was wrong and everyone else has been wrong because they believed him. So I think those kinds of dissections are really important. I think studying the anatomy is really important. And coupling those dissections with the other kinds of imaging techniques we have, like MRI scanning or CT scanning, has really allowed us to get a 3D sense of how all these different bits fit together, which is really exciting.
But I have to ask the guinea fowl and other birds you may have studied. They were presumably not killed afterwards. They went somewhere nice.
They're fine. They're fine. Yeah. So typically these guinea fowl are farm birds, which is really helpful. I think we struggle to do these kinds of experiments with more exotic birds. So for example, we haven't done the kind of X-rom technology, the X-ray things with things like parrots. It's typically difficult to do these. We've done these kinds of X-ray experiments on lots of different animals, but for things that are longer lived, you start to worry about the radiation dosage that they're getting. But for things like farm birds, we're able to do that.
That was going to be my next question about the radiation, the amount of radiation they got.
So it's not too much. So I've been in there. We can do human studies in there. We've looked at people's feet. So it's a very, very low radiation dosage. It's lower than what you'd get at like a dental X-ray at a regular checkup. So it's nothing like even a CT scan or something you'd get like that. Usually the biggest radiation dosage in any of this is the CT scan to get at bone anatomy. But we do want to be cautious. I mean, we take animal ethics extremely seriously. All of our protocols are approved by whole panels of veterinarians. So we really want to make sure that we're confident that what we're doing is okay for the animals as well.
Okay, good. And we have, does this help avoid the shrink wrap reconstruction of dinosaurs?
That's a great question. Yeah. So for those of you who aren't familiar with this, there's this tradition in paleontology of saying we know what the bones of these animals look like. And if we want to make a reconstruction of what that looks like, they'll shrink wrap them. So it's like you've just taken shrink wrap and pulled it over the skeleton. So you get dinosaurs that aren't really fleshed out. There's these really cool artistic reconstructions people have done where they've said, if we just found the skeletons of modern animals, how would we reconstruct those? And so they've created these fantasy reconstructions really of what if you took like a goose skeleton, what would you think that looked like if it were a dinosaur? And you realize how much uncertainty there is between what's preserved in the fossil record and what we see alive today. So I think that's where these kinds of thought experiments, like what if my guinea fowl were a fossil and I only had the bones to work with, how much could I figure out, are really helpful to us because they help us understand what those knowledge gaps are and how reasonably we can infer things about the fossil record from the data that we have. So I think it's really the question that me and my colleagues keep asking ourselves is, how wrong could we be? And I think that's the most important thing to do when you're engaging with paleontology and the fossil record. Like, how much do we know and how wrong could we be? What's that margin of error? So that's why these studies of living birds and living animals are really helpful to us. I think you can make up tons of stories just looking at bones, but this really helps us figure out how much we can and can't figure out.
Absolutely. Yeah, it just makes me wonder if the next Jurassic World, which I think is coming out later this year, is going to put feathers on Deinonychus, or like, the flapping arms and that. Probably not, because they probably want to. But
the public really doesn't love the feathered dinosaurs. I would love for there to be feathered dinosaurs in the cinema, but they're just not as scary when they're cute and fluffy. So there's some great nature documentaries, some great documentaries on Apple TV, on Netflix, some of these more modern shows that have feathered dinosaurs. And I think the kids who are growing up today are seeing those dinosaur shows and they really love them. And they all know that dinosaurs have feathers. So when I get tour groups coming through the museum, it's always the adults who are shocked to learn this. And the kids are like, oh yeah, obviously I knew that. And I know 10 more dinosaur facts than you do, which they usually do. So it's fun.
Well, I sent around to everybody a link for Dino Birds, which was just on was it Nova or Nature? I think it was Nature just like a week ago with David.
Dan Field's work is featured on that. So yeah.
Dan was throughout the whole thing. So that was absolutely fabulous. I do have to say, I know some of you have probably been to the Yale ornithology collections because we've done a lot of tours there. But when you walk in that building and you walk in just from outside and you're like, something feels weird. My like spidey sensors are going, and you look up and there's that Deinonychus above you looking like about to pounce down and on you. And even though it's a skeleton, it's scary. It gets your heart going because you're like, yeah. Like you
don't want to be on the wrong end of it, for sure.
I love the fact that they posed it that way and they put it. It really makes it feel alive, even though it's a skeleton.
Totally.
See who's next. Dawn, I think we answered your question. We talked about husbandry, imagery, and going back to the farm. Get to you in a second. Mimi. Rebecca said, these weird modern animal reconstructions are from early artists John Conway and C.M. Cosman in All Yesterdays. Very worth checking out. Cool.
Totally. Thank you, Rebecca. Yeah, I'd forgotten the name. It's All Yesterday. It's a wonderful book for anyone who's interested in that. It's just really cool to see what you'd come up with if you only had skeletons.
I remember seeing the one with the hippo, and our big Rubenesque hippo is like in the reconstruction. It's like this tiny little weird looking animal that
looks weird thing. Yeah, it's just very strange.
Wonderful. Mimi, you're up. You need to unmute. There you go.
I have that book, All Yesterdays. I was just going to show it.
Oh, wonderful.
To shrink some of the shrink wrapping.
Oh yeah.
Yeah. That's one of my favorites is the whale.
Oh yeah, look at the whale.
A nice sea monster there.
It's really wonderful. It's expensive. It's quite expensive if you buy it, but it is so worth it. It helps you really think about old life and all of the things that the assumptions that we used to make that aren't made anymore. We're trying to get away from them.
It's like if we could go back for 5 minutes and just take a look and go, oh, that's what it looks like. Or just solve all these problems.
Yeah.
I was thinking when you're talking about the Deinonychus claw, it reminds me of the cassowary, which I had the pleasure to be able to see wild ones up close and personal in Australia. And, they also have that sort of terror claw. Yes. And, it would be wonderful if you could do experiments with that. But they've actually been seen slashing with their claw. They rip out guts. So they actually just slash it down and rip out guts as opposed to sort of stabbing. Though it's kind of weird that the Deinonychus claw is so curved, which you wouldn't think would be ideal for stabbing. You'd think it'd be more rapier-like, so you could, do that.
Curve up that way. The other thing, getting back to soft tissue, is that the Deinonychus claw we're seeing would have had a giant keratin sheath over it as well. So that's just a small fraction of the length of the claw, right? There's a whole big keratin sheath on top of it that kind of extends the claw, we think, almost to the ground level there.
Wow.
Probably even scarier than they look. But yeah, I also have the pleasure of seeing cassowaries in Australia, and to consider those differences is just really fun. We have a couple of skeletons, and when you look at the actual cassowary skeleton, you can see now that we understand how the joints fit together and how that dictates these things, how differently their joints are shaped. So you can really start to tease apart these different behaviors by looking at the structure of the skeleton and how that differs across different birds and dinosaurs.
Yeah. Well, if you ever want to do any research on parrot locomotion, if you want videos of how they move everything, I'm sure we can help you out with that. We love to take videos of our birds. So
and I guess you have a tutorial coming up. So yeah, you'll take really good ones too.
Could you get a parrot to walk on a treadmill the way you got the guinea fowl? No. Parrots
I don't think they do it.
They probably do it for like 10 seconds and then go, I've got something better to chew up.
Yeah.
They're way too independent to do that kind of research, but if anyone wanted videos, I'm sure we could do that. And I'll, after me, I'll get the contact information for your colleague, because that would be fascinating as well.
Yeah, I'll definitely send it to you. Yeah, Melody's great. I'm sure she'd love to talk to you all. And she's spent so much time with the lovebirds. She's she can tell you about how hard they bite. I think she's been on the wrong end of that one.
Oh yeah. And we all see our birds climb with their beaks. That's why a parrot is missing a leg, they can usually get around fine.
Well, so it's so interesting. This is why I think talking to people who are enthusiasts about these animals or own these animals is so interesting, because in the scientific community, it really wasn't well known or understood that parrots are capable of this mode of locomotion. That was really big news to a large portion of the scientific community. And we got comments, you know, like social media comments and New York Times article comments from all these parrot owners being like, well, obviously my parrot does that all the time. Like, how are you all just figuring this out now? So I think if we can open these lines of dialogue, we learn a ton just by seeing videos of people's animals moving around, talking to you all about how your animals move. So that sounds awesome.
You know, when you have people who are new bird owners, that's one thing you have to tell them. When the bird is like stepping up on your hand, they'll use the beak, and people think they're going to get bit, that they're about to get bitten, so they yank away when the bird is using it to stabilize the part of your hand and using it to move around. So they're doing it because they feel unstable. But you know, you don't know when this is happening. Is that a bite, or is that I'm holding on?
Yeah.
You know, until you get to know the bird, then you're not sure what they're actually doing.
What the
But
intent is.
then the person jerks away, which makes the perch even more unstable, which then makes the bird either fall or bite or not
stressed out. Yeah.
So, yeah, interesting. If you have any more questions, you can put them in chat or unmute yourself. Your animations were terrific. I just loved it. I
Thank you so much.
Clearly labeled, and you know, seeing how everything moves. You could also, you know, what you might want to do in future talks is put a human knee joint there too, to show how limited, how we don't rotate at all, and how limited our motion is, which our motion really sucks compared to these other animals.
Our knees are really boring compared to birds have very cool knees. So that's another interesting thing. I mean, I work with, and I obviously don't have time to fit into this, but I work with orthopedic surgeons down at the Yale School of Medicine. And so I've talked to colleagues there and other places talking about things like how we do hip replacements and knee replacements in humans. There's definitely room to learn from other animals in the animal kingdom and thinking about other solutions that might be better than the joints we started with. So I think that's something that's becoming a little more common in thinking about joint replacement design is why are we limiting ourselves to trying to recreate the human state when there might be other things that are even cooler solutions that we could give people. If you're giving them an artificial joint, you might as well give them the best artificial joint possible. So what does that look like?
That would be so cool. Can you imagine if you did that? A knee replacement gave you the ability to move laterally. Imagine all the sports people be so all these knee injuries that occur in sports because people plant and then they don't rotate, so they tear. But if you had a bird knee, you would just rotate.
Totally. And so there's always questions of like, is that a good or bad design, you know, because you want to be able to move, but moving too much is maybe a bad thing because you want that stability too. So it's always a trade-off.
Right.
Yeah, I'm thinking that it would be a bad, I have a knee replacement, so I know. One of the things, our problems is we are planned on a 4-footed animal, and birds are not. So we barely have enough. I mean, there's a lot of stuff built into our skeleton. I'm a body worker, and there's enough stuff built into our skeleton just to keep us from falling flat on our faces, because we're really not supposed to be walking on, when you think of how few square inches people actually walk on. And so I think you probably couldn't get away with that due to the fact that we have like 3 or 4 million years, depending on who you talk to, evolution. We managed to stand up, and we all have backaches because we're not supposed to do that. And can you imagine adding lateral movement to that? I work on people whose hips go out of whack, and it's just like, oh, I think that would make a lot of work for PTs and stuff. And after that,
it's so interesting how many it's just interesting how many different solutions evolution has come up with, you know, across these skeletal plans. Like even looking at the kind of knee evolution transition that I showed you all today. A question I got a lot is, well, if like animals like Allosaurus, those big meat-eating dinosaurs, if they had hinge-like knees, then how is that okay? Were they just worse at moving around than modern birds are? But I think that they had really different hip joints and they're standing up much taller. So, your birds are crouched, they're holding their knees up really high. Animals like Allosaurus are holding their legs kind of straight under their bodies the way we do. So there's just different solutions that work together when you piece together the entire animal. But it's fun to think about what those different combinations and solutions are across things and how we might be able to mess with that in different ways to get to different solutions from there.
Well, when that new asteroid hits us within the next 8 years, there'll be all these great opportunities for new things to evolve.
There you go, Amy. Looking on the bright side, I see.
All right.
Is
Do we have
anybody
any?
studying? I'm sorry, is anybody studying like human fossils? I know that very often the skull's all that's left or the teeth, but are they studying the articulation of as many fossils as they can get to for locomotion?
Absolutely, yeah. So I also have work with biological anthropologists. So for some reason, human fossils are usually split off from the rest of paleontology, and they call themselves anthropologists. But I am collaborating with some people who specialize in that, and we're looking at the evolution of foot joints from the kind of ape to human transition, trying to think about how we walk on 2 feet instead of 4, and how our feet are modified for that. And there's people looking at all different parts of the skeleton. I have friends working on shoulder joints across that transition, friends working on hip joints across that transition. So it's definitely being studied too. Yeah, that's why I end up doing a lot of experimental work with birds just because birds are so great to have in the laboratory. We can learn so much from them, but that ends up being broadly transferable to humans and fish and possums and all kinds of different things. So we learn a whole lot about just how joints work in general.
Of course, you could just stop.
Feet would be
great.
Of course.
That's
the best animals, obviously.
Obviously. All right. If there are no other questions, I want to thank Dr. Manafzadeh so much for this talk. It was fascinating. We will be posting this to YouTube, and I could watch this again from the start right now. As video is such interesting information. I want to thank everyone for coming out tonight, and thank you so much for joining us. And I'm going to turn off the recording.